Hello and welcome back to another Future Doc House production. We are currently continuing our journey on this new book I've written. It's right here. There we go. Okay, it's basically Medicine, the Ultimate Guide, Clinical Microbiology, quick review by yours truly, Future Doc House. Continuing. Alright, this is bacteria introduction, continuing from the gram staining lecture in the previous video. Alright, there are exceptions. When we gram stain a bacteria, there are some bacteria that are the exception. They do not gram stain positive or negative. They gram stain poorly, maybe one of these gram stain techniques. But more importantly, you'll see in the vignette that they don't gram stain positive or negative. And then you might think, but it's still a bacteria, not a virus, okay? Because they may want to trick you. Oh, it doesn't gram stain. Oh, it's got to be a virus. No, there are exceptions. Think these 6 exceptions. Chlamydia, mycoplasma, rickettsia, treponema, mycobacterium, legionella. Chlamydia. Why chlamydia? Because it's found intracellularly, not just because it's an STD, but it's found intracellularly. And if it's found intracellularly, the bacteria's cell wall is hidden and encased by the cell, and the gram stain cannot stain the cell wall of this bacteria. Mycoplasm, it does not contain a cell wall. No cell wall, no gram stain ability to stain the cell wall because there is no cell wall. Rickettsia, it's a small bacteria. Treponema, again, another STD. Thin spiral use dark field. So it's a thin spiral shaped bacteria, and in order to visualize this, we use dark field microscopy. Mycobacterium, it absorbs acid fat stain. We will dive deeply into Mycobacterium. It's a big family of bacteria, and it's very important. Legionella, think silver stain. Okay, so if you see silver stain, think Legionella. All right, let's talk about this big mess over here. Before we talk about this big mess over here, let's talk about cell envelopes and the cell wall. Cell wall, cell envelopes, it's made up of, could be made up of an outer membrane, a cytoplasmic membrane, and you've probably gotten all of this in cellular microbiology. But what you know that animals and humans have cholesterol in their cell walls, in their cytoplasmic membrane. Whereas bacteria, they don't. They don't contain cholesterols or sterols. So that means that there's a big difference right there. With that said, let's start with the easy one. And the easy cell envelope, cell wall, would be gram-positive. Yes, gram-positive has less things to talk about. I believe 1, 2, 3, 3 items to talk about. Gram-negative has about 3 things to talk about, but there's more details in some of these. So let's start with what we know. All right, gram-positive. Gram-positive, this is, if you Google any image of the gram-positive cell envelope cell wall, you get something like this, okay? You'll get the cytoplasmic membrane here with these red things called transpeptidase. We will talk about that in detail. You get above that a peptidoglycan layer, so this is kind of moving from inner to outer membrane. And the cell surface protein, so that's the cell surface proteins. And of course a membrane lipoteichoic acid and a cell wall teichoic acid. Alright, lots of glossary terms here, lots of big medical words. You know I love big medical words because I get to explain them. Let's explain them right here. Membrane lipoteichoic acid. It pertains to gram-positive. Okay, so here are your answer choices when you get a question that is regarding a gram-positive bacteria. They're going to say this bacteria contains A, myrin lipoprotein, B, LPS or lipopolysaccharide, C, lipid A, D, membrane lipoteichoic acid. Okay. Now, obviously, the only choice that's pertaining to gram-positive bacteria is the first one, membrane lipoteichoic acid. And if the vignette is describing a gram-positive bacteria, they're going to want you to choose membrane lipoteichoic acid. Simple! Alright, membrane lipoteichoic acid. It pertains to gram-positive bacteria, okay? It has antigenic properties. Now, what does that mean? Antigenic properties are a good thing because they stimulate the immune system. And when they stimulate the immune system, your immune system responds and goes, we have a gram-positive bacteria invasion and we must respond to this due to this probably lipoteichoic acid. Alright, cell wall teichoic acid. Do not get these two confused. They're going to try to confuse you. But the cell wall teichoic acid, again, pertains to gram-positive bacteria, but they are an antigen determinant. Gram-positive bacteria, it's a big family. Okay, how do you separate a gram-positive bacteria from another gram-positive bacteria? Okay, and it would be these antigenic determinants. Okay, and that's what you would use to figure out which bacteria is causing the disease in a laboratory setting, of course. Transpeptidase, transpeptidase, transpeptidase. I believe there are transpeptidases on gram-negative bacteria, but they aren't useful because transpeptidase is used to build a peptidoglycan layer. Okay, they combine. Let's talk about it. These enzymes are for the formation of linkage of amino acids between peptidoglycan molecules. So they help form this layer. Okay, and the general name for this is penicillin binding protein, and these are the beta-lactam antibiotics, the penicillins. Antibiotics that would be used to attack the transpeptidase to prevent the peptidoglycan layer being formed. Okay, and that's why I have it in red because it's very important. We will review transpeptidase over and over again. Now, why we don't care about transpeptidase in the gram-negative bacteria is because the peptidoglycan layer is very thin. It's linear. Okay, does not have a lot of cross-linkings. It's more of a linear kind of joining of the peptidoglycan layer, and so really to prevent this from forming really wouldn't be as effective as preventing this from forming. Okay, and it's not as effective on gram-negative bacteria. So we really usually don't talk about penicillin or beta-lactam antibiotics being used in this fashion. All right, gram-negative bacteria. Let's go over this quickly. Whew. All right, we've got the cytoplasmic membrane. We've got the periplasmic space. Gee, why do we have a periplasmic space? You'll see later. A peptidoglycan layer, which is very thin and very, there's not much cross-linking. Usually a linear linking versus the cross-linking of the gram-positive. Murine lipoprotein. Outer membrane proteins, a porin here. Google an image of this, you'll see something's very similar to this. Of course, you've got lipid A right here attached, sticking out, a core, antigen, O antigen, and these three parts make up the LPS, which is the lipopolysaccharide. And this area is the outer membrane, and of course the cytoplasmic membrane is down here. So that's how it is. Now I wrote here damaged by alcohol in the staining process. So when we stain the bacteria, all bacteria will first stain crystal violet, right? And so this peptidoglycan layer will be violet. But how do we decolorize? How do we remove that crystal violet? Well, the alcohol would damage this outer membrane layer and allow the crystal violet stain of the peptidoglycan layer to be washed away and be able to be restained with safranin. Okay. All right, let's go over the glossary terms, shall we? Murine protein, murine lipoprotein. Okay, now this murine lipoprotein, which is over here, the in brown. Okay, this murine lipoprotein is the glue that attaches the peptidoglycan layer to the outer membrane layer. Very simple. See, this attaches the peptidoglycan layer here to the outer membrane layer here. Okay. And the glue is the murine lipoprotein. And again, why do I care? Yes, it's the glue, but in your vignette, they're going to give you murine lipoprotein, and they're going to ask you, well, let's talk about gram-negative bacteria that's causing the disease. Would this gram-negative bacteria, A, contain membrane lipoteichoic acid, B, cell wall teichoic acid, D, a thick peptidoglycan layer, or C, murine lipoprotein. And we know that murine lipoprotein is the is only pertains to gram-negative bacteria. So that's why we have to learn that. It's annoying, I know. LPS, lipopolysaccharides, again pertains to gram-negative bacteria. And let's take a look here. Yes, there is an O antigen or O specific side chain. Why do we care about the O antigen or O specific side chain? Because they use this in the laboratories as an antigenic determinant. And this is used to, in laboratory settings, distinguish a different gram-negative bacteria from each other. Lipid A, lipid A, lipid A. You will hear me repeat lipid A until you're blue in the face, or red in the face, either way. It's an endotoxin. Endotoxin, not enterotoxin. They're going to try to trick you. That's a completely different toxin. Endotoxin, that's released when the bacteria are lysed, okay? And it causes the fever, diarrhea, endotoxic shock, and it's possibly fatal. Hence, we need to talk about lipid A in great details. Now, be careful. Lipid A, like some enterotoxins, causes diarrhea and fever. Oh my God, it's so confusing, okay? So endotoxin, lipid A, fever and diarrhea is also very similar to enterotoxin, which causes fever and diarrhea. And a lot of enterotoxins are from gram-negative bacteria as well. So actually no, there's a lot that's from gram-positive bacteria. But you'll see, it's confusing. They're gonna try to confuse you with endotoxin and enterotoxin. Gram-negative bacteria. Anything else? Nope. So that's that. If you like this video and you want more of these videos, please click subscribe. Thank you and have a nice day.
Congratulations! You are a medical student. One day, you will be a qualified doctor and your home will be a hospital or a clinic. But for now, your journey begins in the lecture hall. You will listen to countless hours of lectures and spend evening after evening in the library reading page after page of medical theory. And guess what? This is the easy part. The hardest part is retaining your medical knowledge so it's there when you need it. For example, examinations. But don't worry, we are here to help. Meet Jeff. Jeff is a good student. He goes to all his lectures. He says no to parties so he can stay late in the library. But when it's time to take the exams, Jeff freezes. The knowledge that he spent so long learning just isn't there anymore. Now meet Jennifer. Like Jeff, she is a good student. But instead of just reading textbooks late into the night, Jennifer uses technology to help her study. She also watches lectures on YouTube and practices her knowledge with QUPI, a medical quiz app and online platform. Jennifer likes QUPI because it has 10,000 practice questions covering her entire medical course. This means she can practice her medical knowledge from anywhere and in a way that actually prepares her for the exams. Because she is practicing with exam-style questions, when she takes real exams, she'll be ready. Don't be a Jeff. Begin your 7-day free trial with QUPI today.